Well, hello. Wanted to address your applications, especially application 10, in that this is a gene transfer for the bacteria. This is a lab that we were going to do a couple of weeks after the protein isolation, and COVID had other plans. So we will talk about this application now. We'll diagram it on paper, and then when we get back into the classroom, we will actually run this lab. I don't want you to feel like you're going to miss out. We are going to do that lab. And then this question will make a lot more sense when you've got your hands on the lab. So this is going to be application 10, and will help you quite a bit in what we're doing. So application 10, it says, outline the process of gene transfer to bacteria using plasmids, utilizing restriction endonucleases, restriction enzymes, and DNA ligase. So what they're talking about here is a process called transformation, which is the same as genetic engineering. And this is an extremely common lab procedure. It's been around for about 20 years now. So you have your bacterial cell. Here's your Escherichia coli. And you have your genomic DNA here, where bacteria attach their DNA to the plasma membrane. And then they also keep little circles of DNA in their cytoplasm. And this circular DNA, it's called a plasmid. And we utilize plasmids in biotech all the time. It's a very, very useful molecule. It's just a little circle of DNA, unique to bacteria, by the way. So you can remove this plasmid. It's an easy protocol. So much of this biotech is so easy. If you have a thumb and you can do a micropipetter, you can do biotech. So you dissolve the plasma membrane, and then using a couple of filters, you can isolate the plasmid. So what you end up with is a bacterial plasmid. It's just a circle of DNA. And then what you do is you cut this plasmid with a restriction enzyme. In what is commonly called an endonuclease. Very expensive molecules. So when you take this circle and you cut it, it opens the circle. In the lab, this is called nicking your plasmid. You nick it, you cut it, and it takes what used to be a circle and it opens it up. So here is your opened plasmid. Now, what happened over here then is you had a Homo sapien. And in the late 80s, they isolated the genome of human beings. Just out of a cheek swab. It's no big deal. Well, what they did then, late 80s, early 90s, is they pulled just one section of this out. And the gene that they pulled out was the insulin gene. Now, at the time, this was groundbreaking science. This was revolutionary to the biotech industry. And so what occurred is they cut out the insulin gene. Now, what's important is that they used the same restriction enzyme that you used over here. You use the same restriction enzyme with both of your DNA samples, with the same restriction enzyme. All right, so you have a circular piece of DNA from the bacteria. You've taken that circle and you've cut it, so you've nicked it and you've opened this circle. So now what that allows you to do is that allows you to insert or combine the insulin gene with the plasmid. This is literally take a microcentrifuge tube, take your micropipetter, draw up the plasmid, put it in your tube. Draw up the insulin gene, put it in your tube. And then, in that same tube, you mix an enzyme called DNA ligase. And what that does then is it glues or attaches or splices the insulin gene here into the plasmid. This is why it's so important to use the same restriction enzyme. So the end of the insulin gene here and the end of the insulin gene here are complementary to the ends of the plasmid. And so what you end up with is a bacterial plasmid containing the human gene for insulin. Okay? This is called recombinant DNA. If you recall, when I passed around the insulin, Mrs. Mains's insulin, and we saw the outside of the box and it said rDNA or recombinant DNA. That's what they're talking about. This, when you put it inside of the bacteria, it turns the bacteria into a GMO, which is exactly what we're going to draw next. So you take this plasmid and you put it back inside of the E. coli. Same thing, you get a microcentrifuge tube and you put in your cells and you put in your plasmid, and you get the cells really cold and then really hot and then really cold, and the plasmid is able to go right through the plasma membrane and now sits in the cytoplasm of the E. coli. And this step of inserting foreign DNA into the E. coli, that's called transformation. Because you are genetically transforming the E. coli from wild type E. coli into a GMO. So this is now GMO E. coli. Why? Because it's been genetically modified. And how did you do that? You inserted recombinant DNA into E. coli. And we'll do this when we get back into class. You'll see. Well, this cell then goes through its reproductive process called binary fission. And produces clones. Right, and I'm going to be a little sloppy with my drawings here. Here's your genomic DNA. That doesn't have to be very, very special, in all honesty. But what does each cell contain? That's the important part. Each of these cells that's produced from binary fission has that insulin gene from the original cell. Okay, so now all of these cells are also GMOs. Well, these GMOs, they're cells. They read the DNA. They then go through transcription and translation, right? Messenger RNA, ribosomes, and they link amino acids to make a protein. Well, what protein? Well, it depends on the gene that they're reading. So since these cells are reading the human insulin gene, these cells then make human insulin. So this is how, when we passed around Mrs. Main's insulin bottles and you saw that kind of cloudy liquid on the inside, usually that's clear. Well, that is the human insulin that they then take and they put it in a bottle and they sell it human insulin, okay? And that human insulin, even though it's a human protein, it wasn't made by humans. It was made by a GMO. That GMO was produced by first isolating a circular piece of DNA called a plasmid. You nick that plasmid to open it up. You insert the human gene. You then take that human gene, combine it with the plasmid. That's called recombinant DNA. This recombinant DNA is then reinserted into the E. coli. The E. coli then passes those genes, or that gene, to all of the cells that are formed through the cloning process of binary fission. You then get swimming pools full of this bacteria that produce tremendous amounts of human insulin. And this is how Mrs. Maines injects human insulin that was originally made by a GMO E. coli. So that should help you with gene transfer in application number 10.